Okay, I'm here to talk about gel electrophoresis, and this explains the objective that goes over. I can explain how and why DNA is cut and tested. So gel electrophoresis basically is a method that we can separate DNA, RNA, or any kind of proteins. We're going to focus on DNA since that's what we did in class, and it's separated by molecule size. The smaller the molecule, the faster and farther it will move down the gel. We're going to talk in a minute about what happens here. There's the DNA sample. It's taken out, and we're going to talk about what needs to be cut from it. And then the DNA is going to be placed into these wells, just like we did in class. After it's placed into the wells, it will be placed in a buffer solution and then into a chamber where it will eventually give us a nice looking gel like this. So we'll go through this process. The first thing we need is to cut the DNA. It needs to be cut because it's super long. We do not need to test all of it. So we use restriction enzymes. Restriction enzymes are added right to solution. It cuts the DNA in specific spots. There's many different restriction enzymes. They're all specific to a certain gene, a code it's looking for. So it might be looking for GATTCC on one end and then a CA on the other end, and it cuts that out. And then anything in between, it will cut out. So this happens in a test tube. DNA is cut, then it's small enough that we can actually run it. So how did we prepare the gel? This is what we did in class. We took an agarose and buffer solution. The agarose was a solid. We weighed it out. The buffer was a liquid, and we put it in the microwave and we heated it up. From there, we poured it into a tray, which had a comb in it. The comb was there just to make sure that the wells did not close, so it was there to make an opening. We then took the comb out when the gel was solid. We kept it for the next day, and then we finished it by taking the DNA samples. We used 8 different samples, and we loaded them into the gels using a pipette. So this is what we used. If you notice, we had this black line right up here, and we had a red line right here when we loaded our gels. And that black line was to represent the negative or the cathode end. So this gel would be put in so that the negative end is closest to the wells. The positive end, or the anode end, which is why it's red, was on the opposite end. So we took this gel, we placed it in the buffer solution, we put it with a power supply, which I'll show you in a minute, and after about 15 minutes or so, we started to see the DNA fragments form. And so this shows you just different lines in different places. We did it using color. This is what the box we used. This is the black line that the wells were by. This was the red line. These are the combs that we used to make the wells inside of the gel. And the 2 different size, depending on what group you were in, gels went into this box. I hooked it up to a power supply, and it sat for about 15 to 20 minutes. This was our result. As you can see, we used color instead, and color is just like DNA samples. It separates based on size. So the red must be a smaller size molecule because it moved farther and faster. So we saw that this first DNA evidence, which was in well 1, produced red and blue. The second DNA evidence just produced blue. And then you can see all the way down the different colors that were made. We then compared this to a known sample, and were able to figure out what piece of evidence, which were these 8 wells, came from what suspect, or what if it came from the victim. In real life, scientists don't use colors like we did. They actually use a solution, and they do put a dye in it, and then they need to use a UV light in order to see it. So the solution they use is not something they can touch with their hands like we did for us. So what they create is something like this. And you're going to see different bands as well as different thickness of bands all the way down the slides. This can be used for paternity tests as well as to figure out if a suspect committed a certain crime. And that is what we talked about with gel electrophoresis.